It is my distinct pleasure to introduce Dr. Mokler. He is a professor of pharmacology within the Biomedical Sciences Department of UNE COM, and he also serves as chair of the Biomedical Sciences Department. He got his bachelor degree in psychology, followed by his doctoral degree in pharmacology and toxicology at the University of Michigan. He then...
Michigan
State. Oh, no, I knew I was going to do that. Fair enough. I'm going to read it now. And then he did a NIDA postdoctoral fellowship at the Medical College of Virginia. He came to UNE in 1986, which means that he's had just over 30 years here. He has been a very vibrant member of this community, not only at COM, but at the university itself, and has promoted not only education, but really wonderful opportunities such as interprofessional education opportunities for the students here. And in his spare time, he has had a vibrant and extraordinarily active research program here. He has been focusing on behavior and neurocircuitry within the brain, primarily focusing on serotonin, as you'll hear about today. He has over 70 papers, and he has collaborated with people at places like the Harvard Medical School, see, I messed up, now I can't even catch my breath. Harvard Medical College, Michigan State University, got it right that time, Boston University, the University of New Hampshire, as well as Temple University. So he's had a long, distinguished career as a researcher as well as an educator, and it is with pleasure that I'll turn the microphone over to him. So please welcome Dr. Mokler.
Thanks, Tamara. Thank you very much. Thanks, everybody, for coming. This is very heartening. So first off, I'd like to point out some special guests. Everybody here is special, but some very special guests. First, I'd like to point out the Dr. Mokler, my father, Corwin. So he goes by Moke, okay? And if people want to know, I'm Mokey, all right? So, but he doesn't like Mokey. He thinks that sounds not so good. So, yeah. A couple of other people here that are very important to me. Chuck Landry, who was in my laboratory back in the 90s, says a little bit about how old he is. Chuck is like the original UNE graduate. He went undergraduate here, he went medical school here, he did his residency here, and now he practices neuromuscular medicine up at a clinic in Manchester with Doug Jorgensen as his partner. And then also as a surprise today, another one of my undergraduate students, Jessica Paddock, showed up. So that was a great surprise to see her here. Thank you both for coming. It means a lot to me. So my mentors, certainly my father. I've got a picture here from a couple of years ago, back when I actually had some color in my hair still. It was still a little bit red. And he had a few more hairs, maybe. Also, Dick Reck, believe it or not, you know, these days usually you think, okay, I need a picture for a seminar. I just go out and type in Dick Reck and I'm going to get a dozen pictures of this guy. I couldn't find a single picture. You know, I went out and asked one of the graduate students that was with me in the lab. He didn't have any pictures, so we couldn't come up with a picture. John Rosecrans, up here in the corner, was my mentor at Medical College of Virginia. And then Peter Morgaine, who was here with me, he actually started in '85, the year before I started here, and he was here until his death in 2010. And he got me involved with the prenatal malnutrition studies down in Boston. And he and I worked together and published together for many years, and I do count him as one of my mentors. And then my students, okay? One of the things I want to do in this particular talk is to sort of underline the involvement of my students in this work, okay? There's Chuck, there's Jessica, John Nadeau. Was John Nadeau here when you were here? I can't remember. Anyway, he just started in general surgery at Southern Maine Healthcare, so he's a new surgeon over there. Many of these people have gone on to professional degrees, occupational therapy, dentistry. Some of them are lab technicians, DOs. You know, a lot of these people are DOs. Here's some more of the students, and I'll bring these up as we go through each of the individual topics that I want to cover today. And even more of my students, okay? Going forward, we do have some fun in the lab too. This is celebrating, I don't know what we were celebrating there, but springtime probably, okay? I think this is the spring lab party that we had at the house. We had quite an exciting group there. So what I want to do today is I want to do sort of a few vignettes. So I'm not going to tell a really long, involved story about a particular aspect of serotonin, but a number of different vignettes of the research that I've done over the years. Starting off with looking at the role of serotonin and behavioral effects of hallucinogenic drugs, which was my thesis dissertation. How does serotonin involve with prenatal protein malnutrition in the brain? Looking at some of the techniques using in vivo microdialysis to assess how the brain is wired. Okay, what is the circuitry in the brain? How does it functionally interact? Then a short period of time where we worked on a project looking at the pathology of in sudden infant death syndrome and how serotonin was involved with that and making big advances there. And then a particular study that we did looking at how serotonin and norepinephrine and dopamine differ in the brains of rats, sort of as a baseline, looking at some of the different chemistry there. So when I first started looking at graduate schools, I was really fascinated with the ability of chemicals to alter consciousness. Okay, this is the 70s, you guys, okay? This isn't the 70s, all right? Yeah, I know a lot of people were interested in that too, yeah. And so I looked around and I said, okay, where is the best psychopharmacology, the best neuropharmacology places in the country? And one of the places, and I think, Dad, you had a friend, I've forgotten his name, but he's right, he worked in the same department with you, that you went to him and said, okay, where should he go for pharmacology? And the two places that he pointed out was Michigan State University and Medical College of Virginia, and Boston University. Okay, those were three places he pointed out. I've ended up working in all three spots. Okay, so I ended up at Michigan State. Now I did my undergraduate there, as people talked about, but I was out in San Francisco on the West Coast and doing, actually, believe it or not, I was a banker out in San Francisco. And so I came back to Michigan State and started to look at the serotonin role in hallucinogens. And Dr. Reck and Randy Commissaris, who's a good friend and was in the lab at the same time I was there, they had worked up this behavioral assay where they could give rats different drugs, different psychoactive drugs. And by the way that the rats were behaving in this operant response, they could tell whether they'd gotten a stimulant or they'd gotten a sedative or they had gotten a hallucinogen. And so we went back and because they were, LSD is related chemically to serotonin, mescaline and DOM are related to amphetamines. So how do they affect behavior using serotonin antagonists? There was a number of different hints to suggest that they might be working through serotonin. And so they had developed this behavioral assay. And then we started to look at how we could give non-selective and selective antagonists against serotonin to sort of tease out what might be the involvement there. So this was one of our first studies that we published, and this is looking at a variety of serotonin antagonists and their ability to antagonize this behavioral effect of the hallucinogens. And the two hallucinogens we were looking at here in the top slide here is LSD, and in the bottom slide is an amphetamine called DOM. Okay, it's a amphetamine-related compound. And the behavior was these number of 10-second pauses that the animals would have as they were working to get food reward. And this is very positively associated with a drug that has hallucinogenic activity. And if you would look in the cages when these animals stopped responding, okay, so if at about 200 micrograms of LSD or 400 micrograms of LSD, this is a fairly whopping big dose of LSD. And if you'd look into the chamber while these animals were in the chamber after giving it, they'd be just sitting over in the corner, sort of just staring off into space. Now, I can't tell if an animal is hallucinating, okay? How do you tell if a rat's hallucinating? I did try. But I did not do anything like take a hallucinogen myself and ask them whether they were hallucinating or not. I figured that that was going overboard, you know? I believe in introspective pharmacology only to a certain amount. So basically what we saw is as we gave these different types of non-selective serotonin antagonists, that we were shifting this dose-response curve, and that now required higher doses of LSD, and actually very large doses. In fact, if you look at here, which is the effect of metergoline, which is a very non-selective serotonin antagonist, it pretty much blocks the effects of DOM completely at any of the doses that we gave. We just didn't push it out any further than that in order to look at the response. Now, this was an exciting time in the history of serotonin research because in 1979, just a few years before I did this work, Peruka and Snyder at Johns Hopkins University did this study looking at multiple serotonin receptors in the brain. So there was some other evidence suggesting that there were multiple serotonin receptors in the periphery, particularly in the gut, that people had looked at before. But these were now, this is the first study looking at the binding against serotonin receptors in the brain. And they call these two subtypes of receptors the 5-HT1 and the 5-HT2 receptor. And they were looking at, as you can see, differential binding of tritiated 5-HT, tritiated LSD, and tritiated spiroperidol. And it turns out that LSD binds to both, 1 and 2. Spiroperidol is selective for 2, which is interesting because this is an antipsychotic medication, which Karen knows a lot about, and it was binding very selectively to these 2 receptors. So around the same time, when you're a pharmacologist, what you do is you wait for some medicinal chemist to come along and make a new compound that you can use to play around with. Okay, so you're always saying, okay, now we have these 2 receptors. We need a drug that we can use to play with it. All right, and it didn't take long for them to come out with this compound, perinpirone, which was a selective serotonin 2 antagonist. So we jumped on this and started to put it into our assay to see what would happen. And in fact, when we give different doses of perinpirone against LSD, and now we're looking at mescaline as another subtype of hallucinogenic drug, we saw a very large shift, again, in the dose-response curves for these particular drugs, and indicating that the serotonin 2 receptor was intimately involved in the effects of these drugs on this behavior. They have a lot of different other targets, and as we've gone along, we've dissected it out a little bit further to where we now have serotonin 2A and 2C receptors Those of you that know serotonin research know that there's 15 different serotonin receptors at this point in time, going from 5-HT1 to 5-HT7 with a lot of subtypes in between. At this point in time, where we're at, and it's hard to get funding for this kind of hallucinogen research, so there's not a lot of people that are doing it, but mostly it's selectively 2C receptors that these hallucinogenic drugs are working at. Okay, now to shift to the next story. So this is sort of the bigger story of the research that I've been doing over the years, because this goes back about 20 years now. I'm looking at prenatal protein malnutrition. And up in the upper thing here, you can see myself, and here's Peter Morgaine. This is Jill McGahey, who's at University of New Hampshire, has been working with the behavioral assays with us for a long time. This is Doug Rosine. You can recognize this. This is in PICUS. Doug Rosine, who's at Boston University, does a lot of the neuroanatomy. And then, of course, Peter Morgaine again. I didn't seem to have a picture for Janina in here, but she's at Boston University. She's been the PI on the project for a number of years. And she's pretty unique in the sense that she's an MD psychiatrist, adolescent psychiatrist, and she has developed this project and this interest in prenatal protein malnutrition. And she works with us on an animal model, but at the same time, she's down in Barbados working with a group of patients that were identified at birth as having been exposed to prenatal protein malnutrition. And they're now in the third generation of looking at these offspring. And we've definitely seen effects in the first generation that was directly exposed to the malnutrition and the second generation. So you'll see that this develops. So here's some of the crew that has worked on this prenatal protein malnutrition over the years. So there's a number of different studies that are very key to our understanding of what's going on with the malnutrition. So the Dutch famine study was looking at humans that were exposed to this malnutrition soon after the Second World War. There was famine in the Netherlands, and those patients that survived, the offspring, have been looked at, and they've been seen to have an increased risk of depression. There's also a Chinese famine study. There was famine in China. I believe it was in the 50s and 60s. And they've been looking at those patients, the children that were born to mothers that were exposed to the malnutrition there. And they see an increased risk of schizophrenia. And in Barbados, Janina has found with her studies that they have an increased risk of depression, but also has attentional problems, okay? So if you do any kind of behavioral task looking at attention, these patients show difficulty in paying attention. It's not really what you would call ADHD, but it's sort of the similar symptoms. And these people actually have severe attentional problems to the point where they're pretty much disabled in their ability to work. So this is one of the papers that's come out that Janina did from her work in Barbados. So with our animal model of protein malnutrition, we look at the offspring of these animals that are exposed to the malnutrition. We assess them behaviorally with a number of different tests. Then in one subset of animals, Doug Rosine and his group will take and do the neuroanatomy, and they will look at things like CFOS and 2-deoxyglucose, which are, CFOS is an immediate early gene, shows neuronal activity. 2-deoxyglucose shows metabolic activity in the brain. And they're able to look at what areas are activated during the memory tasks that we're working on. My group would then look at the in vivo microdialysis, which I'll explain in just a second here, to assess the activity of the monoaminergic neurons. And then we also have a group down in New York City looking at epigenetics and what are the epigenetic changes that are seen there. So this is a technique that I developed and have worked with over the last 30 years. I really started working on this technique when I first got here. And it's basically taking a probe and putting it into a rat brain. And this is about 5 millimeters long. And you can see a very small probe at the end. The width of this is about on the order of 0.5 millimeter, 500 microns wide. So you can put it down into a rat brain, and this is a coronal section of a rat brain. And this is the prefrontal cortex, which is an area that we're very much interested in. And then we put an artificial cerebral spinal fluid down through this and across this membrane, and if there's any neurotransmitters out here around in the extracellular fluid, it gets picked up, okay? A certain percentage of it gets picked up, and then gets transported back out of the dialysis probe, and then we can inject it on an HPLC and analyze it for the amount of serotonin or dopamine or norepinephrine that's in that particular sample. So we're able to do this in a conscious animal as the animal's walking around. We can have them doing a behavior at the same time. We can give other drugs either peripherally or through the probe in order to assess what's going on. We can compare groups of animals, controls versus malnourished animals. So one of the first things that we wanted to do, and this is back in '98, and this might have been some of the stuff that Doug and Jessica worked on, was to show that this was really neuronal release, okay? And so briefly, if you give tetrodotoxin, which I think everybody probably is familiar with, it's from the pufferfish, it's a sodium channel toxin, it's very potent toxin, scary to be working around it. But if you give it through the probe and you start it right here, you can see that the amount of serotonin shuts off almost completely. Okay, there's only a little bit left. Another way to slow down neuronal release is by taking the calcium out of the cerebral spinal fluid, okay? And then you get a depletion of calcium around those neurons, and no longer do you get the neurotransmission. And it comes down quite a bit, not as much as with TTX, but you can see it goes down. And then one other way to stimulate release of neurotransmitter is to increase the amount of potassium that's in the fluid. Now, these are in conscious animals, okay, that I'm doing this. And we're in the dorsal hippocampus, an area of the brain that we were looking at the time with our malnutrition studies. And you can see here that you get a very large 10-fold increase in the amount of serotonin that's being released if you put the potassium chloride into the cerebral spinal fluid. So we felt that this indicated that we had a good amount of evidence here that we had neuronal release. So what happens when we do microdialysis in these malnourished animals? And this has been a consistent finding. This builds on previous studies. There's probably about 4 or 5 other papers looking at serotonin levels in the brains of these malnourished animals. And you can see that there's a significant increase in the amount of serotonin in these malnourished animals in the dorsal hippocampus. What the reason for this is not entirely clear. I'll show you in the next study. We have a little bit of an idea there. Now, in this particular thing, we were mixing up different types of way of stimulating the systems, okay? And we were actually putting electrical probes in to stimulate the median raphe nucleus, which is a site of the neuronal cell bodies for serotonin that project up to the hippocampus. And so when we stimulate, you get a brief stimulation over a period of about 20 minutes, and you can see that there is a significant difference in how much serotonin is being released, and the malnourished animals actually decrease significantly. Although this is a percent of baseline versus the total amount, so if you were to look at the amount, you might actually be seeing something different there as far as the percent decline is larger, but the actual amounts are probably pretty similar. So we went on to look at other significant things. So here we wanted to look at 30-day-old rats, so young rats. And here we're in the dorsal hippocampus, and we get this increase in serotonin, which we've seen a number of times before. And now if we give a chlorodiazepine epoxide, which is a benzodiazepine, which is a sedative and an anti-anxiety drug, you see that immediately you see a very significant drop in the amount of serotonin that's being released. So I'm thinking in terms of the fact that this could be very well a stress response, that these animals are stressed, so you see a large increase in serotonin. And increases in serotonin in the hippocampus are oftentimes seen with stress responses. So that was one of the things that we were working on. Now, we've started to shift our focus away from the dorsal hippocampus. We weren't seeing a lot of behavioral effects that we could relate to the dorsal hippocampus in our studies. So we now shifted away over to prefrontal cortex, which makes a lot more sense in my mind, given the linkage to depression, given the linkage to schizophrenia, that now you're going to be looking at what are these things happening in the prefrontal cortex, and also with the attentional problems that the patients have. So again, this is the same group now, and we're looking at deficits in attentional set shifting task, which is an attentional task that we have the animals do. And then we were looking at the metabolic activity in the prefrontal areas. And we saw decreases in the metabolic activity in these prefrontal cortical areas. At the same time, we were seeing these deficits in attention. Okay, so there's a decrease in metabolism in the prefrontal cortex. In another study, this was actually one where we combined some animal work with some human data. So this is pretty much talking about the human data as far as the title is concerned, but in this paper, we also had data from some of our animal studies, and we showed that there was a reduced performance in another attentional task if the animals were distracted. The malnourished animals showed some distractibility there, and also saw decreased brain metabolism in the prelimbic area of this prefrontal cortex in this particular paper. So at the same time that we're doing all of this work in terms of metabolism and attention, we're also looking at the animals for their release of serotonin. So this is one of the early studies that we did looking at prenatal malnutrition in the prefrontal cortex. Now I'm shifting a little bit and including dopamine. So the technique that we use when we're doing this microdialysis, we're able to pull up this cerebrospinal fluid, and it not only contains serotonin, but also it contains dopamine, it also contains norepinephrine, it also contains a lot of other things that we're not analyzing. But the analytical system that we can use allows us to look at the norepinephrine, dopamine, and serotonin. So we're starting to look at the correlations between all of these things. And in this situation here, when we looked at the medial prefrontal cortex, we actually saw a very large decrease in dopamine. Stress did not have too much of an effect here. This is the actual dopamine levels. This is the percentage of control. So standardizing it to a certain percentage before we produce the stress. And you can see we didn't see changes in serotonin in this particular study, but we did see the decreases in dopamine that occurred. Going a little bit further now, in another group of studies, another cohort of animals, we're now in the medial prefrontal cortex. Now we're starting to look at a different parameter, or a different set of parameters, in that we're not only looking at these 3 different neurotransmitters, but we're also looking at both sides of the cortex. Okay? I think anybody that's had Neuroscience 101 knows that there's laterality in the cortex, all right, in our behavior. So there's some aspects of our right brain that are different functions than our left brain. So it only makes sense that the chemistry may be different when we do this. And so for a long time, as I've done this work of looking at different areas of the brain, I've always said, okay, if we're just doing one side of the brain, we always stay on one side of the brain. Okay, we don't mix it up. You're either right side of the brain or left side of the brain. So now we're starting to put 2 probes in to measure simultaneously 3 different neurotransmitters in both the left and the right cortex. Okay, same areas of the brain. And you can see there's significant differences. Okay, these are basal levels of serotonin. And in the right, we don't see any effect of malnutrition. But in the left, we see a large increase, as we've seen in other studies. All right? Dopamine. Dopamine is suppressed right less than left, and then malnourished even more so than the well-nourished animals. So very low levels of dopamine. Similarly, very low levels of norepinephrine in the malnourished animals. Well, low levels of dopamine, low levels of norepinephrine are considered to be part of the process of attention, or inattention for that matter. So these animals have deficits in attention, and we're showing that, at least in the right side of the brain, in the rat, that there's low levels of dopamine and low levels of norepinephrine. And I've started to use this laterality index to sort of capture that information. And you can see that the well-nourished rat is very different looking than the malnourished rat in terms of the balance of neurotransmitters. Now, as far as I have been able to determine, nobody is looking at the behavioral aspects of left versus right brain in the rat. So we need to have that work done. There's a challenge out there. I'm sure there's tons of money at NIH for that work. You just have to convince them. So in summary then, looking at all of these different studies that we've done, we've seen that we get a fairly consistent increase in serotonin in brain areas, although I'm not presenting data on orbital prefrontal cortex, which is another area that we've looked at. But we see increases in norepinephrine in both sides compared to well-nourished controls, but no changes in dopamine and serotonin. So because we're not seeing these global changes in these neurotransmitters, there's got to be some kind of rewiring that's going on. It's not a deficit that's occurring like one of the nuclei has been lesioned, okay? It's got to be something else. It's got to be some other reprogramming of the brain, which is one of the focuses that we're having. Norepinephrine, dopamine related to the inattention. Serotonin tends to be related to impulsivity, but we really haven't looked at impulsivity in these rats. Okay, so now shifting again, all right? So always interested in what is the functional interactions. How can we use this ability to probe into the conscious brain to look at functionally how these different areas of the brain are operating? And 2 areas that are very important in aspects of serotonin work is that there's 2 nuclei in the brainstem for serotonin that project up into different areas of the forebrain. And that is the median and the dorsal raphe. And these are both midline nuclei. This is the dorsal raphe that sits right here. And then there's a very thin sheet of cells going right down in the middle here. This is the median raphe. And they project up into the forebrain. And so what we wanted to do is to look at these 2 areas and see how they interacted. So we basically did probes into both of these 2 areas. And in one set of studies, we injected glutamate, which is an excitatory amino acids, would stimulate the release of serotonin in the brain. And then into the dorsal raphe and monitored the median raphe. And then controversially, we infuse glutamate into the median raphe and look at release in the dorsal raphe to see what the connectivity is between those 2. And we got a pretty interesting result in that if we give a high dose of glutamate into the dorsal raphe, we see large increases in serotonin there. And then it follows that we see large increases in the median raphe as well. So there's some kind of connectivity between those two nuclei. There's an excitatory connection. And we've done the studies to show that this is not due to diffusion of the glutamate away from the dorsal raphe into the median raphe to cause stimulation. Because if we do the reverse and now put glutamate into the median raphe, dorsal raphe doesn't respond. Okay, so we can use this technique to probe. Other people using the same technique have done things like put a probe into the dorsal raphe, put a probe up into the frontal cortex, infuse glutamate into the dorsal raphe, look what happens up in the frontal cortex. Okay, and so you can probe around into different areas and draw a map of excitability. So I'm looking for about $10 million over about 20 years, and I might be able to get that work done. Mapping it out. Okay, so this is now an interesting story. This is a very, very interesting and productive side product that came out of the work that I was doing, and it was directly responsible from Jill Hoffman, right here, who was an undergraduate student here. And she worked in the laboratory. I'll show you some pictures of the work that she did when she was here the first time. Upon graduation, because of her work in my laboratory, she got a job at Dartmouth as a lab technician. And she was working in the lab of Bob Darnell there, and he was looking at respiration. And he was part of a large group that was studying the respiratory system in the midbrain because they were interested in what was going on with children that die of sudden infant death syndrome. So he was connected to this large group. The PI was Hannah Kinney, who was down at Boston Children's Hospital. So this is how these large groups of scientists sort of mix and move around together to produce amazing results. So because of the microdialysis stuff, Bob Darnell and I started talking, working with Jill. He sent me some samples to analyze from the studies he was doing, which was actually in pigs. And I started going to meetings and was trying to see if I could fit into this program project that they had. They had too many investigators. They didn't have room for another one. They were splitting it in too many ways. But Hannah called me one day and she said, listen, we've been doing this work on the pathology in these children that have died of SIDS, and we want to know if you can measure serotonin in postmortem tissue. And I said to her, no, you can't do that. Okay? Number one, you know, if somebody dies, the brain starts to fall apart right away, everything's decomposing, you're probably not going to get anything measurable out of this. And then I said, but let me think about it, okay? And so I went back to her and did some research, and actually there's some old studies that somebody did back in the 70s where they took rats, okay, this is a little gruesome here, okay? They took rats, they killed them by asphyxiation, and then they laid them out at room temperature in the lab. And then at different time points, they would take the brains and they would look and see what happened to this postmortem tissue, to these neurotransmitters. And in fact, serotonin doesn't go away over a period of a couple of days, okay? Dopamine actually goes up a little bit. It's hard to say why it goes up a little bit. Norepinephrine stays pretty simple, so I said, or steady. So I said, okay, but you know, and she said, that's okay, we'll have ways of doing some matching with controls. We have a way of balancing out, you know, if the time of death, if we know when the time of death, because, you know, these children die in the middle of the night. You don't know how long it's been since they actually stopped breathing. So I said, okay, let's do this. And the postdoc in the lab at the time was Jody Duncan. This is another, this is Elizabeth Andrews. okay, who was an undergraduate in CAS here, and she went on to get her Ph.D. University of Hawaii. But Jody Duncan was a postdoc from Australia that came up to work with us, and we did this study. Okay, we took these brains. It took a long time because it was, as the brains became available, we would do the work that we needed to do on the thing. Thanks for coming, Jeff. Yeah, good luck. From this work came out a publication in the Journal of the American Medical Association. This was part of a lot of other information that was there in terms of it wasn't just what happened with the serotonin levels in the brains of these children, but there was autoradiography that they were doing. So there was a lot of backup material. And this was actually the second publication, the second cohort, where they were focusing in on serotonin. So the serotonin neurons that are in the midbrain, in these areas, this is a gigantocellularis. In this area is one of the places. The paragigantocellularis lateralis, which is right here, was one of the areas we sampled. And we also sampled the raphe obscurus, okay? And the serotonin neurons in these areas of the brain respond to CO2, okay? And as all the medical students know, CO2 drives respiration. You get an increase in serum CO2, you increase respiration, all right? So the serotonin neurons seem to be intimately involved. Here's just an indication that Jody did of where the punches were taken. So there's your raphe obscurus. Here's your paragigantocellularis lateralis. And when we looked at the serotonin levels, this is a previous study done by David Patterson and his co-workers at the same group in 2006. And you can see this is 1A receptors. Okay, so it's just gone. Okay, versus the control infants. All right. And then if we looked at the raphe obscurus in our SIDS babies, we saw decreases in both areas, significant decreases, compared to controls as well as hospitalized controls. Okay, so this gave us more evidence that there's a deficit in the serotonin neurons in this brainstem area that may be, and the current hypothesis is that these neurons are failing to mature in a timely manner. And the children, eventually, if they survive these apneic episodes, they will go on to develop normally. But if they get into these apneic episodes, then they can possibly die. So at this point in time, I know that Hannah's still working on this project. Her goal before she retires is to come up with a treatment. I'm not quite sure what the treatment might be. It might be a serotonin reuptake inhibitor to bolster up the serotonin that's in these areas of the brain to continue the respirations. It's hard to say. Okay, and then finally, the last little vignette to share with you is looking at the adolescent brain. So there's a lot of work being done right now with the adolescent brain, looking at the adolescent brain and maturation of the adolescent brain, because this is the last area of the human brain that matures, okay? And it's involved with decision making. And so the National Institute on Drug Abuse is very interested in this area because it may have some explanation as to why kids use drugs, okay? That they have less of an ability to make the decisions to inhibit the drug use in order to, not use the drugs. So this is Amanda Stady, and Amanda came into my office as a freshman undergraduate, and she told me she was going to do research in my lab, okay? Didn't ask. She said, I'm going to do some research in our lab. I don't know what I'm going to do, but I'm going to do research in our lab. And so she and I worked and talked about, okay, what's interesting and what are we doing right now in the lab and what could we do? And over the next 4 years, she and some other students in the lab that worked with her did this paper. So this is her paper. This is her study. And so what we did is we took rats, young rats, adolescent rats, we took adult rats, we took male and female rats, and we put probes into the right and left prefrontal cortex, and we looked at the release of these neurotransmitters, okay? And the results are, again, interesting and hard to explain. So this is it. And you can see the female adolescents and the female adults, pink, yellow, blue for the male adolescents, green for the adults. I don't know why I picked those colors. Pink, obviously, and blue. But so serotonin is very different. The male adult has fairly low levels in comparison to either the female adolescents or the female adults. The male adolescent actually has somewhat more than the male adult. Dopamine looks a little bit different. We see that in the left prefrontal cortex, we see some differences between the female adult on the left versus the right that we don't see in the adolescent females. And the males, again, have the lowest level of dopamine in both sides. And norepinephrine was very variable, and we didn't see any real strong indication of one way or the other here. Again, there's differences in these brains. Left versus right, male versus female, adolescent versus adult. And we did do some studies looking at stimulating with methamphetamine, which is a stimulant. It really didn't go very far as the results from that. It didn't show clear differences between different animals. So that's pretty much the story. Looking at laterality, we saw some differences in the same thing. We still don't know why we see these differences, but they're definitely there. And we've wanted to put these into some kind of program in order to sort of investigate what these differences are going to be. So over the years, this is the support that we've had: NIMH, NIDA, NICHD, the National Institutes of Health. First Candle is a SIDS foundation that has supported some of the work we were doing. NASA and STEM research supported some of the other student work that we had done. And finally, I'd like to thank my family, because they were there through the whole thing. And be glad to take any questions that you may have. Thank you. Yes, Karen.
Oh, it's not working.
There you go.
Congratulations.
Thank
That's
you.
a great body of work. As you know, I'm fascinated by mood and metabolism, and I wanted to know, on your protein malnutrition study, did you and your research team, or you and Davidoff, look at how these changes in neurotransmitters might predict glucose dysregulation, which we know happens with protein malnutrition?
How long, Amy, have you tried to get those animals to look at exactly that? Yeah, that popped into Amy's head the first time we talked about it, and we tried for years, and we just never could get the actual animals, because, you know, we raise hundreds of those animals, but we use them all, you know? And so I still got some hearts somewhere in a -80 freezer, Amy. If you want to go after that.
So we did talk about it.
Yeah, we did.
It didn't happen.
Thank you. Yeah.
So I kind of wanted to broaden the context on the epigenetics a little bit more. And there's good evidence that the phenotypes that you see due to epigenetic changes actually carry on through multiple generations, as Janine is like F2, F3. And I know you didn't get into real details about this, but you were just doing protein malnutrition as opposed to calorie malnutrition, right?
Right.
And the other question, one, maybe just explain why, but also that, because doesn't it reflect back to the Barbados group? They weren't calorie deficient, they were protein deficient.
Right, exactly. So that's the focus on the protein malnutrition as opposed to calorie
If you
deficient
said it, I
malnutrition.
missed it. But did you ever take it more than one generation out?
Actually, we tried. That was our last grant that was not even discussed. What we wanted to do is we actually had it set up so that we were going to look at multiple generations. And the one study that I showed that was biological psychiatry, that actually shows the epigenetic profile. And one of the tricks about the epigenetics is, what do you look at? You've been interested in Oglicnac, okay? You can't even pronounce it. Oglicnation. We were looking at histone methylation. Totally different. How many different things can you look at? And we've saw a lot of changes in some key genes when we did that. And we've also seen some important changes in the epigenetics of it's not epigenetics, but epigenetic changes in the DNA in the rat model as well. Okay, so we wanted to do that with our last grant that we put in, but like I said, it sort of fell flat for a lot of different reasons. I don't know why. It was multi-generational and it was very expensive, and trying to figure out exactly how you were going to raise all these animals over multiple generations was crazy. It was crazy trying to do that. Yes, sir.
From the emergency department standards, I'm thinking in comparison to Narcan, do you see any practicality or any interest in controlling the effects of LSD or some of these neurotransmitters more acutely in terms of reversing a patient coming to the ER, say, high on LSD?
Yeah, there are some studies looking at the using some of the non-selective serotonin antagonists to block the effects. There was an ED study done using one of the antagonists in patients coming in presenting with LSD. It didn't show that much efficacy in terms of, other than the standard protocol was to put them in a quiet space, in calm space, and keep them quiet and calm. Yeah, and at the time that they were doing that, they had many more people coming in with PCP and ketamine, which you can't even touch with these drugs. So, it was hard to delineate exactly what they're coming in on in order to do that study. So that was a good question. Derek.
Have there been any studies of SIDS, the incidence of SIDS with infants of mothers who are on SSRIs?
That's a good question, and I don't think anybody's really gone out there and looked at that.
No, antidepressants have been a real issue in general for outcomes.
Yeah, and they are. There is an issue about using antidepressants in pregnancy, especially some of the SSRIs. There are some outcomes, but nothing that relates to the SIDS issue. Now, there is a lot of correlation with tobacco use, alcohol use. Those increase the risk in these children. Any other questions? All right. Thank you very much, everyone.